So hi everyone, let's start the class. Yeah, am I audible? Hello?
Yes, ma'am. Yes, yes.
Yeah, okay. Yeah, thank you. I'll just share this. My slide is visible?
Yes, ma'am. Yes.
Yes, ma'am.
Okay. Yeah. Last day we saw some questions, right? So today also I'll be discussing one of the assignments. It's the second assignment, which is from previous year. So I'll be asking one question. You can answer it, and then why is that answer is correct or others are not correct. We'll have a discussion regarding that. Okay. So let's see. What is the question? Is that microarray data was collected from a wild-type Arabidopsis plant. If you're new to this class session, you know that Arabidopsis is the model plant. So, and the mutant plant lacking lateral loops. So here they're talking about two kind of plants. One is the wild type, and another is the mutant. The mutant is having this lateral loop lacking. So they're asking that what could be concluded from the data. So first question is the differential proteins in the mutant plant. Second is the differential post-transcriptional modification. And now the third one is the differential expressed mRNAs. And last one is the none of them. So which one is the answer for this? Yes. What is the answer for this? Why no answer? Are you guys able to hear my voice?
We can hear, but there is some background disturbance.
Yeah, that's because I'm in lab right now.
Okay. But it's manageable, no problem.
Okay. Actually, I'm expecting. Okay, I'll try to speak louder if that works.
That's fine, that's fine. You're audible. Yeah.
Okay. And I'm surprised that I'm not getting any interaction from the other side. Okay. Fine. So anyway, the answer to this question has nothing to do with what kind of plants it has been taken. The answer to this question is only one sentence or only one word here, that is the microarray data. So you just have to know what microarray does. That is the main point. If you know that, you can answer this question. So why the answer is C? Let's have a look, and why not other options? That also you can have. So next slide is it visible? Okay. Fine. So microarray is but the name is coming from a plate, or which has an array, means an arrangement of different uh, what I call it spores, suppose. So multiple spots are there, suppose in a plate, and in each spot you have different label DNS. Okay? And so what happens that you have the cDNA library already. So cDNA is what is cDNA? I'll tell you. So when you have your DNA in your genome, that DNA gets transcribed to mRNA. So from mRNA, that is through reverse transcription, that leads to cDNS or the complementary DNA. So and in many of the model organisms, they have this library of complementary DNA, so that we know that what are the transcript population in a specific in model organism. And why the cDNA is used? Because cDNA and compared to DNA, if we talk about, so you know what, that when a gene can have exons and introns. So exons are the one which get transcribed into mRNA, but introns do not. They get excised. After mRNA, so that after further splicing, that introns get out and only the exon in the mRNA. Okay. So but why we're not dealing with mRNA is that the cDNA or the DNA itself is much more stable than RNA, so it's easier to handle cDNA. So that's why whatever experiments is done is basically done with the cDNA. So now what you are doing is, so you have your normal cells. One second, I will just admit someone. Yes. So they what they're doing is that tagging the cDNA with a specific probe. Suppose here in first one they are tagging with. Suppose in the first one they're tagging with GFP. So your the cells, the mRNAs from the normal cells are GFP tagged. Suppose the other one in the cancer cells, they are red fluorescent probe. So they will show some color under certain specific question. So what happens is a very basic thing, that you are collecting the sample, you are collecting you're doing the RNA isolation, then you are reverse transcription, through reverse transcription you are forming the cDNAs, and then what you are doing is you are mixing both the population of cDNAs, and you're loading it in each of these spots. Okay. So what happens, the spot has already what do I say this, the already sequenced some, so a sequence of cDNA already present in this spots for individual genes. Okay, so when you are adding this fluorescent tags, what's happening, they're binding to this complementary strand, and that's how they're giving one signal. So signal is like that, they would be green or they would be red, but here the combination of a signal is working. So what you can see from here, that if that specific RNA is cDNA is not present in the cells, then it will not show any kind of fluorescence. So you will not be able to see any signal there. But if it is present in both the cells, why it is yellow? Because your green and this red color both together are giving rise to a yellow signal. Okay, but if it is present only in the normal cell, what will happen? That the normal cell, this transcript will only be able to bind to it, but they will not. But the red fluorescent probe, they will not be able to bind to it. Okay, so and that's why you will only be able to see the green signal from here. So vice versa, you will see only the red if it is present only in the cancer cell. So what happens that in normal cell compared to cancer cell, if we talk about, there will be a variety of different pathway activation and all. So because of which there are always, so that RNA profile will be different from the wild type. So there will be thousands of probably transcripts which are upregulated or downregulated compared to your normal cell. Okay, the other healthy cells, if we can talk about. So it is possible, suppose for one specific gene, a specific mRNAs are getting upregulated. So upregulated means that it is present more in this. So suppose it is present in healthy cells, the specific transcript is present in number 10 suppose, and here if it is present in 100, then the signal which you will be able to see will definitely be red, right? So if from here it depicts that this cancer cell actually harbors that gene. So here what will happen, this each specific spot you already know is specific for which of the gene. mRNA. Okay, is this clear from here?
Yes.
Okay, thank you. So, from here you can easily understand that this is actually for the differentially expressed mRNA. So, differential expression is the term used for that when you are comparing with something wild type and mRNA is young, right? So, in the mutant, that's why. And definitely not, this is not for protein, as we can see it's specific to RNA, and definitely post-transcription modification also comes with protein itself, so after the translation that happens. So these two options are ruled out. That's the third option is the correct answer. Okay. Let me add with some. Let's move to the next question. So the reverse transcription polymerase chain reaction used to. So we know this reverse transcription polymerase chain reaction, if we it short, is the you all definitely aware of it because of COVID, that the RT-PCR result used to come, used to see all these things, right? So let's see what is RT-PCR. If you have no idea till now, we can have a look into it. So what could be the answer for this? Please try to answer this. Meanwhile, I'll admit. This is disappointing that no one is actually interacting. Anyway, fine. If no one is answering, I just assume that everyone is able to hear it and see it clearly. Okay. So, what is RT-PCR? First, let's see what it is. Then we will try to answer. So, as the name suggests, that reverse transcription polymerase chain reaction. If you're already familiar with polymerase chain reaction, that is what happens, that you amplify. You try to amplify a specific DNA, right? Specific gene sequence. So, what it does, that you design primers. So, suppose this is your. So suppose this is your DNA sequence, and you have you design primers against it. So forward primer and reverse primer, and then use of polymerase enzyme, and along with some conditions, what it happens that it's extend the sequence, right, along with annealing and other thing. So what happens that your DNA gets amplified in each step. So each step it gets doubled, right? So if you have one copy, so 2 copies, 2 to 4, 4 to 8, 8 to 32, and it becomes so big that it becomes easy for you to detect. So, and it's not only for detection, but it is used for any kind of cloning work. Cloning work in the sense that you want to some kind of work you want to do. Suppose you have a specific gene X in your plant, and you want to see what you want to see the protein, you want to purify the protein from it. So definitely you have to clone that in some vector that will be having this the protein expression efficiency, along with the vector, along with it should have better compatibility with some strain. So and then you express the protein, and then purify, and you go to other. Yeah. Please mute if you have turned it on. So that protein purification, it can be used for other protein works, or any kind of, if you want to characterize the protein, or any other kind of work where you want to study it could be something Western blot, HTS, or any other protein related one. So that's why the cloning part is very important. So for cloning, the first step what is done is basically to amplify that sequence from your genome. Okay. Yeah, that's what. And depending on your target, it could be cloned in different vectors. It could be protein-protein interaction, or you want to see the transcript, whatever it is. You first need to do this. So, but what is What is real-time PCR is? Real-time is that in the real time, you can say, or the live, you can say that how much is the change of a transcript. So basically what is done is that if you remember about the COVID, what we were doing is that compared to a healthy or uninfected cell condition, we were comparing that how much viral RNA is present in that, right? So there is something called CT values and all that I will come to that. So first let's see the principle of it. So what happens that you have something called Tachman probe. So it could be other probes as well, something inside the brain and all. So anyway, just remember this Tachman probe. So what it happens is that it binds to DNA and it has some fluorophore molecules bind to it and a quencher molecule. So when your forward PCR and the reverse PCR starts amplifying your DNA, or the extension if you can see, then what happens that due to its extension, this part gets cleaved off, the fluorophore molecule. And because of the cleaving, this fluorophore molecule is now separated from the quencher molecule. So what was happening earlier is whatever specific wavelength you were giving, it was emitting some wavelength, and that energy was actually quenched by this molecule. But now, as this fluorophore molecule is separated from the quencher, it can no the emitted wavelength or the energy cannot be quenched by quencher anymore. That's how it leads to a signal, and that signal is measurable. So not measurable, I mean that can be measured. So that's how suppose once you're getting the signal, and with each cycle this signal gets amplified, so that leads to so the signal actually measures passively that how much cycle it might have gone through. Okay, so if you just imagine, if I say that it will be detected, suppose the signal will be detected once 100 of cycles gets completed. So what will happen that? So that means you have to go for 100 cycles. Now imagine you have two sets of cells. One is the healthy, another is the some infected cells. Okay. Now, if your healthy cells contain suppose one transcript and your unhealthy cells contain 10 transcript, so you can understand that to reach to 100, your healthy cell will require 100 cycles. Suppose, okay, and suppose in each cycle you are getting only one signal. So, but in the unhealthy cells, what will happen? You have already 10 transcripts, so it by 10 cycles, within 10 cycles, it can reach to 100, right? So healthy cell will require 10 seconds to detect, to come to a position where signal can be detected, but unhealthy cell will require only 10 cycles to get detected by the machine for a signal, right? So that is something is known as threshold value, or the cycle threshold, CT value. So what happens is that suppose you have different samples. So just imagine this one is the healthy, another is the unhealthy sample, or the infected sample. So what happens that in one of them you may be getting the signal less below CT, less 10 cycles, while in the other case you're getting a higher cycle threshold number. Okay, so that shows that this curve, or the sample which is showing this curve having harboring more transcript compared to this, that's why you are able to detect the signal at an earlier time point, right? So if you have checked the CT values while your RT-PCR results have come, you might have seen that lesser the CT values, it's actually not a very good condition, right? Is it clear now? So from here you can say that so the reverse transcription is, so RT-PCR is used to detect the presence of transcript in a tissue, right? And and definitely not for the spatial. Spatial distribution is not done in this case, because here what happens is you're taking the whole tissue, and then whatever the the experiment is in vitro method, because you're isolating the RNA, making the cDNA, and then you're going for this experiment. So definitely so whatever, if you want to see the spatial distribution, it has to be in vivo. In vivo means that inside the tissue or inside the cell. Okay. And another is the quantified protein content. Definitely it's not related to protein, as it is talking about the transcript. Another is study the chromatin structure, no way. And so the answer is direct the presence of transcript in a tissue. Okay. Yeah, and it's not only for the comparison whether that specific transcript is present or not, that also can be checked. Yeah. If you're getting in the signal, then it's good. Okay. Is it clear? I really want an answer for this, otherwise how can we move forward? Yeah, can I have an answer, please?
Yes, ma'am, it's clear.
Thank you. So let's see the third question. So which of the following is an example of targeted suppression of gene expression? So the question, if you read the question properly, it's actually hidden in the question itself if you already know about it. So they are asking about which of them is an example of targeted suppression. So targeted means that when it talks about targeted means it's specific to some gene. So here, so the gamma ray. So gamma ray is actually a mutagen, if you know about it already. But it can lead to double stranded break or single stranded break in your chromosome, and that because of that break, what happens that if some break happens in your chromosome, and let me come to the slide better. So just concentrate in this figure. Let's suppose there is any kind of gamma radiation is you have treated your cells with some kind of radiation, and that's how there is some kind of break. So it could be single stranded break or double stranded break. So what happens that after this break happens, you will say, yeah, there will be some repair system that will come into picture, and they will just repair it. Of course, they will repair it, but the problem is they will often lead to some kind of mutations. Okay, so the repair is actually error prone. Because of that, there could be some phenotypic effect in that. But what problem with this gamma radiation is that it can actually go randomly anywhere in the bases, and it can cause that break. So it's not specific to anything. You cannot. Suppose you want to target your gene X, but you cannot do it because it is actually unbiased. It can go randomly and do the job. So this is definitely not a targeted DNA. Targeted suppression of gene. Okay. So next, see, we all know about EMS mutagenism, right? Ethyl methylsulfonate. So ethyl methylsulfonate. So what it does is, so you know that we have 4 bases: A, T, G, C, adenine, guanine, cytosine, and thymine, right? So what happens that A always pairs with T and G always pairs with C. So A, T, and GC. But what EMS does is that EMS leads to some at the 6th oxygen position, it leads to the ethylation. So it results in the O6 ethyl guanine, and this O6 ethyl guanine now can pair with thymine, which actually it was supposed to pair with cytosine, but now it pairs with thymine. Because of this, there is a shift from GC to AT, because you will see that this thymine, when it will get replicated in the next step, this thymine will actually bind. It will pair with adenine, right? So initially, which it should have been GC, it will now become AT. Right? So this kind of, and we know that a single nucleotide sequence can alter the whole gene expression. It can lead to different aberrations of transcription or anything. It can lead to incorporation of stop coders or any other mutations, which altogether may lead to a different kind of protein which might not be functional. So, but the problem again is that EMS mutagenesis is not specific. If you treat cells or any plants with EMS, it can lead to the mutation throughout the genome anywhere. Okay, so it's a random mutation. Another is the tDNA mediated insertion. tDNA, if you already know, that it is actually the nature's own genetic engineer. What it does is that in the agrobacterium cells, bacteria, it contains a TI plasmid. Okay, the TI plasmid contains a tDNA. So this tDNA, after infection to the plants, it can actually go and bind to the. It gets inserted into the genome of that specific plant. Okay. So, if you already know about it, agrobacterium tumefaciens led to the something called gall, right? Gall formation, because along with TI plasmid or the tDNA, they have other enzyme coding genes like as auxin and cytokinin, I guess. So what they does is that they leads to the higher proliferation of that specific organ, and that leads to formation of gall. But the gall formation is not something we want or desire. So that's why nowadays what is being done is that it's being disarmed. Disarmed means those genes are being excluded, and only the tDNA portion is being kept. Okay, but again the thing is that tDNA can go and insert anywhere in the genome. It's not specific to any genome. Okay. So again, that's the random DNA insertion, right? So the thing is here, if you see this picture, you have a specific gene. No, sorry, it's any gene, suppose, not specific, and then your tDNA fragment with left border and right border, this portion is getting inserted into it. Okay. Here, if you're wondering what is NPT2, it's actually a antibiotic selection marker. So what happens that you have to, so when you dip, this is a dipping method, floral dip method. So what is happened that in this beaker you will have the solution where you have the agrobacterium harboring that specific gene. Okay, so you will dip the plant's floral buds in this, so that this agrobacterium can actually enter into the germplasm, because flowers will have sperm cells and egg cells, right? So if it can go and attack the germ cells, then it can get inherited to the next generation. But the thing is, it's not always 100% foolproof method. That's why some of the plants will be having this construct, but it's not necessarily. So that's why for the selection of that, there's something known as selection marker that is being used. So if it is an antibiotic selection marker, so if you're growing the seeds of this next generation in this antibiotic selection marker, if they're harboring this marker means that it can actually catabolize a specific antibiotic, so it can grow. If it does not have this specific antibiotic, it will perish or die. So the one that leads to actually makes it easy to select for the plants which have actually got the construct of it. Okay, I just explained because as I mentioned, the whole process. Anyway, so then you can later on with the you have to map which region of your genome is actually got got the insertion. Okay, so but the problem again, you can see here that it is a random DNA insertion. So neither EMS, neither gamma ray, neither tDNA is specific. It is actually all a non-specific gene targeting method. So the last answer which we're left with is the RNAi. So is that so what is this RNAi? RNAi stands for RNA interference. Term itself that it's talking about interference. So something, it has to interfere with the RNA, right? So what is happening that in the cell, in the nucleus, you have DNA that's giving rise to your mRNA. So mRNA then gets transported into the cytoplasm, and then from the mRNA, through the process of translation and ribosomes, it leads to the formation of proteins, right? But what if you stop this translation at what if you stop this translation somehow? So what you can do is that we just read about the AT base pairs, like ATGC. So DNA and RNA all have this property of binding to someone, complementary binding, right? So if you can actually, so this is known as sense. So if if this is known as sense mRNA. If you can design something which is antisense, or you can say the complementary to the mRNA, then it can go and bind to your mRNA, and because of this binding, it will hamper, it will actually hamper the translation process of it, and then it can lead to inhibition of translation. That's how it's actually repressing the function of the gene. So you will know, because the majority of the cases, I mean not majority, you can think that final product of the DNA is a protein, right? So it's not always protein, because we know that RNA also has some function, but even if RNA has some function, it is getting hindered. So so it's not leading to the full formation of protein, or if it has any other RNA related work. The important thing here is that you're targeting a specific RNA here, because to target this RNA, you should know the sequence of this RNA or the DNA, so that so you can specifically target the gene X or Y. So this is known as RNA interference. That's how we are targeting specific, or the targeted suppression of gene expression, right? So that is, if I have put the slide, there's something known as silencing RNA, that also similar. It works with some complex that can lead to the degradation of the RNA itself. Okay, is it clear?
Yes, ma'am, clear.
Thank you. So here is something about RNA in situ hybridization. So as we are talking about real-time RT or the RT-PCR, so RT-PCR we saw that the presence of some transcript in us in a tissue, but that was not specially, we cannot see the expression of exactly where in the tissue it is being expressed, that we
And your
cannot tell.
slide is not visible.
It's not visible.
No, ma'am. On the previous one you have shown it till now. Yes, ma'am. Yes, ma'am. Now it's visible. Yes, ma'am.
Okay. What about now? Is it fine?
Yeah, it has moved to next one. Yeah.
Okay. So please answer something for this. Otherwise, we'll let it be an interactive class. That's what I mean. So I'll read the question, you answer for it. Next, so what is it saying that during RNA in situ hybridization, proteinase K is used to enhance cell permeability and remove nucleases which may otherwise degrade the transcripts. So at what step proteinase K treatment is done during in situ hybridization? Okay, you can answer it later. So the idea is to know what is this RNA in situ hybridization. So if we talk about in situ hybridization, so the term itself is in situ, means something has to do with the in vivo condition. Okay, so if you saw that real-time PCR which I was talking about, is that in that you are taking the whole tissue and seeing whether the transcript is present or not. So it's a yes or no question that is being answered or addressed. But here what is happening, that in a specific tissue, exactly in which position, okay, that is present. So it gives you and spatial information. Along with it can also give you some temporal. So just think about spatial information is positional information of a specific RNA in a tissue. Okay. So if we read about it, so as it is also saying in situ is there, as it is also talking about hybridization, means that it has to, by hybrid is something, is a mixture of two things, right? So it has to bind to something. So what can you do? Like the mRNAs, whatever is present in your cell is definitely not visible to you, right? If you remember the microarray which I was talking about, there were also something called labeled the fluorescently tagged CDNAs, right? So by checking that tag, you can actually visualize the other RNAs present in that. So here what they are doing is they are actually doing the similar kind of thing. They are tagging a probe means which will bind complementary to your target mRNA, and then they will check for the they check for the signal. So it actually varies. It kinds of varies the kind of signal depending on the what kind of tag that specific probe has. It could be chemiluminescence, it could be luminescence, anything. Okay, but majority of the time it requires confocal or chemiluminescence kind of technologies. So let's see what it is done basically. So basically you are preparing your tissue. So there are some processes in which you might be uh, you have to go through some process to prepare your tissues with hydration, dehydration and all. So then you have to isolate the RNA, and the important part is that you have to make the leveled mRNAs or the cDNAs which can bind to the RNA. No, not cDNA, it is mRNA itself. And if it is RNA, then it has to be RNA itself. And then if they are binding and they are along with your substrate incubation, if the product formed is giving some kind of signal, then it can be measured through microscopy or any other technique needed. Okay. So if you see, if you have a look about the little bit of detail of that. So the labeling, first of this labeling is that label your DNA RNA probes and oligonucleotides in one step direct method. So whatever is you are tagging your labels. Okay. Then hybridization is here you're binding your mRNA with a specific probe, and then blocking to reduce any kind of background binding and all, and then you are detecting and then you are analyzing the tissue according to your own techniques. Okay, so that's how it is being done. So here question is a bit more in the process of it, how it is done. So if you have heard of proteinase K, then you might already be knowing that in many proteinase K is actually that degrades protein as well as it degrades nucleases. So nucleases in the sense that can degrade DNA, but the RNases. So you know RNA is something very unstable. So unstable in the sense that the RNase is an enzyme that degrades RNA in a very rapid rate. So what happens if you have gone through any kind of RNA isolation and all, you will know that you are not even supposed to talk while you are isolating the RNA, because even our saliva contains RNA, so that can degrade the RNA molecules which you are actually isolating. So we have to take precautions, we have to be vigilant for that. And so for that, we are actually here dealing with RNA, so we have to treat with proteinase K so that it does not degrade, as well as it removes any unnecessary proteins which might be interfering with the binding. Okay, so that's why. So from here question you can pretty clearly say that as RNA is your first step, then definitely it has to be done proteinase. So hybridization is the main thing we are looking for, right? So if we want to want our RNA to be stable and free from any other background work, then it should be leveled with proteinase K pre-hybridization. Okay, so that's an obvious answer is the pre-hybridization. I hope it's clear. Let's move to the next question. Please ask if you have any kind of doubts or anything. Okay, as I already spoke about it, RNA in situ hybridization. I'm expecting that I'll get this answer for this. Any cases, please, I will give you 2 minutes of time. You read it and please try to have an answer. If you're not able to tell it, please write it in the chat box. Yes, any answer, please? Is anyone there in the class?
So we don't use any RNA probes. So that should be D, which is false there, right? We usually use the probes which are DNA probes only. RNA probes won't be used.
Hmm. Okay.
So it should be the ribo probes have identical sequences to target RNA is the false among the given options.
Yes, yes. Yeah. So the answer 4 is the D is the correct answer. The reason is very simple here because they're saying that riboprobes have identical sequences to the target RNA, but it cannot have identical sequence to the target RNA. It should be complementary. Otherwise, it will not be able to bind that RNA. So that's why this is answer D is the answer. Actually, it could be. I think it could be for DNA. Sometimes it can be used for DNA, RNA, DNA in situ also, some kind of, or DNA, DNA. I'm not very sure about it, but based on my knowledge, I can say it looks identical as it cannot have identical sequences. So this should be the answer. And answer C is saying that the probes can be leveled with either radiofluorescent or antigen level vessel. That is very true. It could have different kind of levels. And this is very true. That is based on the complementary binding of a nucleotide probe to a specific target cell, DNA or RNA, as I said. It can be both, and it is used to detect nucleotide sequences in cells. Yes, so we can also check for a specific tissue that whether that specific sequence or the specific mRNA is present or not. So definitely these all are the correct answers except the last one. So let's see the last question. Sorry, not last one, number 6. It says that which of the following will be the best strategy to study the subcellular localization of a protein in plant? Yeah, please. I'll wait. So sometime let's read it. Answer. Okay, what's the answer for this question? Best strategy to. So I will give a hint here. They have already talked. It's already in the question. The answer: subcellular localization of a protein. So definitely, the answer has to be um, answer B. That is a translational protein reporter fusion. It cannot be promoter because even though your promoter is active, it does not necessarily mean that it will give rise to a protein. And most important, and not only that, the protein which is getting translated might not be necessarily it will be present near the promoter itself. It can go definitely it will go outside of the nucleus to get translated into protein. So definitely it will never match with the promoter position with the protein, right? So what happens that in the nucleus, DNA leads to formation of RNA. RNA goes to the cytoplasm and that leads to the formation of proteins. So that protein can later localize in the nucleus or in the membrane or any other organelles. That's a different thing. But for that we have to track the protein itself. Okay. So next is Western blotting. Western blotting is again done with the whole tissue, so it cannot detect the subcellular localization. Subcellular in the sense that in the cell, where it is in the membrane, cytoplasm, nucleus, or any other organelle as such. Okay. So let's see what is this. So this is something called OSIG1 is a GFP protein. So it's tagged with the GFP protein. This is like this some rice protein probably. So that is being tagged with your GFP. So you can see the GFP here. This experiment is done in protoplast. So protoplast is the isolated. Protoplast is generally isolated from the plant tissues. They form some spherical shape. So is it used for in vitro as an in vitro system in many cases. I mean, not exactly in the plant where you are taking out some part of it from the plant. So, so then what is happening that you have this GFP, something else signaling is done. There is a DAPI. DAPI stains the nucleus. So if both this GFP and DAPI, if they are co-localizing with each other, then we are saying that this protein is actually localizing in the nucleus. So that's how it's being confirmed that this specific protein, with the help of this reporter, is localizing in the nucleus in a specific time. It can the thumb proteins can actually move from here and there, so it might not be always in the nucleus. So the thing is, okay, next question is that in order to discover specific cell type marker, the scientists generated a collection of translink lines with random insertions of a promoter-less reporter gene and screen for specific reporter activity in the domain of interest. So what is this technique called? I am sure you have already attended classes on this. So sorry,
Can you please project that slide?
I have already projected that slide, but is it
But
not
we are
visible?
still in sixth question. Here it is sixth question which is shown.
Okay, maybe we'll give it some time, it will come. Yes, I'm able to see now.
Yes. It's seen. Thank you.
Yeah, maybe because of the internet issue. Okay. So, yeah. So, you might have already attended, sessions regarding this question, but if you could answer, it will be really helpful. What about others? Only ma'am is answering, no one else is answering.
Actually, it is marked there. We can answer it easily.
Okay. Okay, that's fine, but at least someone who can explain it then. That would be nice.
Yeah.
Why you think that is the answer? Oh, it is actually not in the slideshow mode. In the slideshow mode. Fine. Yeah, so the method is known as promoter trapping. So why it is known as promoter trapping, let's see. So from my knowledge, promoter trapping is basically it could be different kinds. So first kind is that what you are doing is that suppose you have a gene of your interest, and you know the sequence of the gene, you know the promoter of the gene. Now you want to know that what could be the other transcription factors that might be binding with it. So what happens that transcription factors, they are proteins, right? But they are less abundant compared to other proteins. So sometimes it becomes difficult to detect them. So if you have heard of chip, where what is being done that against a specific protein, you raise the antibody and then you pull that protein, and along with some DNA sequences come, and you see that where this protein was binding in the DNA sequences. That one thing. But here, it is actually the exact opposite of that thing. If I say from my concept, that you were actually expressing that promoter, and then you are seeing what are the proteins it's binding to it. So that is one kind of promoter. It's actually basically should have been the transcription factor trapping. So anyway, that kind of experiments is done. People do that. But if you see here, what they're talking about, that here is saying that a promoter less reporter gene. So you have this reporter gene in the sense that gene along with your reporter, it could be GFP or any other thing, and then he was checking for the expression or any enzyme you can think of it for any kind of assay, and then you are randomly inserting this reporter gene, the vector which will having this gene. So what will happen? The vector containing this promoterless gene, reporter gene, it will get incorporated into the genome. But as the thing is, it will not have any promoter. It will not get transcribed. So to get it transcribed, it has to come in contact with some kind of promoter. That's the idea. 1 second. So so for that, what you are doing, so you are seeing that checking the reporter. So if that is being expressed, it could be some kind of antibody markers also. If that is getting expressed, that means it has come in contact with some kind of promoter, and then later on associating or checking the sequence of that promoter and all, we can actually identify which promoter is that. So this kind of studies are basically done for the identification of some new promoters or the putative promoters of a specific gene. So I have a new gene, I don't know that what else genes it might, what else promoter it can actually regulate. It can be regulated by what else promoters. So for that, this kind of studies are being done. Okay. So this is known as promoter trapping, and this is talking about gene knockdown. Absolutely no reason that it could be the answer. Gene trapping, as similar kind of mechanism could be, but here they're talking about as it's a promoter less, that means definitely that targeting the promoters. And enhancer is something called enhancer elements that they search for. So probably the principles could be similar, but the targets are different. So let's see, 9th question. Is the slide visible for question number 9?
Not yet. Yes. Yes.
So I actually have to go back then interviews. So which of the following is I think it's already, if it is already showing, so from next year I'll just remove the tick mark, but still you can just tell me which is the answer. Okay. So which of the following is the incorrect statement? Anyway, so SAGE is something known as serial analysis of gene expression, and DNA microarray I have explained previously. So SAGE is something that tags your cDNA, and then it amplifies cDNA along with some other tags, and then they check what are the sequences present. What are the cDNAs present. So the important thing about is that it does not require any prior knowledge of your cDNA library, whereas DNA microarray needs that you already have the information of that specific cDNA, then only you can make the spots with specific cDNAs. So both of these are actually global gene expression analysis tool, not tool, as experiments. But the difference is that it does not require prior information, it requires prior information. And the thing is that both of them are actually revealing the information regarding global mRNA expression level for different tissues or different samples. But the thing is that the second option is the studies have indicated that gene expression mesoforms are highly inconsistent with each other. But that's not true, because both, even though there would be some principal difference, it's actually telling about the global gene expression profile of it. So it should align with each other. And the third one is a stage does not require that. That's what I just told. It does not require prior knowledge of transcript. And DNA microarray can only detect the gene spotted on the microarray. Yes. So your gene should already be spotted in that microarray. Microarray means that plate should contain all of the cDNA in that. So in this way, these two are actually varying. And one other thing is that stage is basically considered as that not that is not that expensive compared to microarray. And the last question is this IST2 hybrid assay. So IST2 hybrid assay, last time, I think in the last class, it was opposite question, that how we do detect the protein-protein interaction. So how will you identify the protein-protein interaction, by which experiment? So the answer was IST2 hybrid. So here actually this. So you already know about it. So let me just explain a bit of it. That what happens that IST2 hybrid, as the name is suggesting, is yeast. So means the experiment is performed in yeast. 2 hybrid means you will have 2 different kind of molecules which you want to study. So here the study for IST2 hybrid is a protein-protein. So means that 2 protein A and protein B, whether they're interacting or not, to know that. So what's being done is that some transcription factors. So we know that a transcription factor will bind to a gene, and because of its binding, it leads to the expression of the gene. So the expression of the gene can be used as a readout for the binding of the transcription factor to the transcription start site, you can say, or the upstream element of the gene. So what happens that in the cellular system or the in vivo system, that proteins does not work alone. They often work in concert. So multiple proteins come together and then perform a specific function. So sometimes it can also be like a key. So a protein could be very important or crucial for a specific turning on of a specific gene. So even though two proteins are there, it requires another protein to come and bind. Then only the transcription will start, something like that. So for that, inside the system, protein interaction is very important for any kind of functional aspect. So to check that whether it happens or not, sometimes it happens that in vivo system could be difficult sometimes. So people generally check it in the in vitro system as well. And of course, there could be certain questions always that cannot be answered in the in vivo system. So sometimes it is taken as the in vitro system. So if we're talking about plant system, two protein coming from plant, then the in vitro system for plant will be yeast. So what is that being done? That a specific transcription factor and a specific promoter is already chosen, and that specific transcription factor is split into 2 parts, and then each of the protein A and this protein B attack with this specific part. And then what happens that if the transcription factor is already split, it will not be able to transcribe that gene. But if these 2 protein A and protein B, which are attached with 2 halves of this transcription factor, if they come together, then it brings the 2 parts of the transcription factor also to each other, and that leads to the formation of a complete unit and leads to the formation, the transcription of the gene, the reporter gene, and that's how it can be selected. Their cells, and this, or not only that, and the growth of because suppose that reporter gene which is being expressed is crucial for the growth of that yeast. Okay, so through observing the growth, it can actually tell whether 2 proteins are interacting or not. And we know the transcription factor has 2, basically a protein has 2 parts. One could be the activation domain, and another is a DNA binding domain. So this, when it is being split, it is actually split into this activation domain, and one is a DNA binding domain, and these 2 proteins can be tagged with 2 different halves of this, and that's how the system works. Okay. Yeah. Of course, there are other method which is talking about here DNA protein or RNA protein. For that, some other is only being used, but there are some other names called. So is one hybrid, is tri-hybrid systems. So let's see if someone is trying to join. Yeah. So CRISPR, you all know that it is a the question here being used is that why CRISPR technique is something which is a gene editing tool, a genome editing tool, but it's actually a family of DNA sequences derived from viruses in the genome of a bacteria. So CRISPR, we use it as a tool for gene editing, or a system for gene editing tool, but it's actually, if we think of the, or talk about the origin, it's coming from some sequences of the bacteria. So it is known as that clustered, regularly interspaced, short palindromic reports. So the term itself here is saying that clustered means there should be some cluster of genes, and regularly interspaced means there might be some regular spaces between them, and short palindromic reports. And palindrome, we all know that which can be read the same from both the sides. Like we talk about mom, M-O-M, mom. If we read it from left to right, that would also be mom. If we read from right to right, that would also give rise to mom. So that is known as palindromes. So how it is coming, so it's bit of immune system for bacteria. So what they do is that, so whenever they get infected with some kind of virus, so what they do is that if they're able to defend that virus successfully, they just not only defend the virus, they also takes or cuts a specific part of the viral genome and then integrate that sequence into its own genome. So, and so it's like a photograph. Like we have killed an army, and let's take a photograph of army and keep it with us, so that in future anyone coming like this, anyone shows this kind of face, we'll immediately know this is the enemy, and we can we should not trust it, and we should actually defend it for the future purpose, we will say. So all the immunity things are basically for the future purpose, right? We think about antibody in our own system, that we produce antibodies and memory cells which can later on recognize it and fight against it. So here it is the same. So but the thing is, it actually does it in a very organized way. If I say they have a specific loci called CRISPR locus that contains different so suppose it depends, actually it will depend on the lifetime or the kind of viruses it interacts. So it could be it can vary from each bacteria to bacteria, like what kind of CRISPR sequences they have. And so if some bacteria might have depended 10 viruses in their life, so they can have 10 different kind of that lineage can have 10 different kind of gene cassette, but while others can have even more or even cannot have any. So that this kind of this kind of things and variable things could be there. So so what happens that the different viral genome sequence, the part of the viral genomes along with the spacer, they form a cassette. And then what happens that they with the help of some endonucleases, something like Cas9 or other Cas enzymes will also be there. What will they do, that they will so when some virus comes or infects the bacterial cell, they will form this RNAs they produce will be known as guide RNA. So this guide RNA along with some endonuclease, they will form a complex like as here. And as the guide RNA sequence will be complementary to the viral sequence, they can go and bind to the viral sequence, and then the endonuclease enzyme, which will be here, is the Cas9, that can actually cleave the viral genome, and because of that it can defend the virus in the future. So this is the original history, or the talk about how it works. So yeah, so that's how it works in general. But what is being done right now is that what scientists are doing to manipulate it for humankind or any other other targets or any other scientific studies that they are actually. So if you want to target any gene of a specific strain, then you can actually design the guide RNA against that gene, and along with that endonuclease expressing vector, then so that the whole complex can go and bind to that specific sequence, the which you are targeting, and that can cleave. And as I've already mentioned, that if when some cleavage happens or break happens, that it tries to that specific system will try to repair it through its repair mechanism, but it will lead to some error prone uh, incorporation of some nucleotide that might give rise to the knockout or knockdown of the gene. And not only that, it could be different enzymes which can actually modify that. It could be some enzyme which can lead to the separation of that specific sequence, maybe interacting through other transcription factors, or it can lead to the activation of. So these are modifications which can happen. And you also know about the methylation status, as I was telling in the last class, that it can also modify or alter the methylation status, and that can lead to the activation or inactivation of a specific gene. Okay, so yeah, CRISPR, we also talk it as a gene editing tool, but it's a family of DNA sequence that is basically derived from viruses that bacteria initially use. It's a natural system for bacteria, which is being used for scientific purpose nowadays. Okay, is it clear?
Yes, it is. Thank you.
Yeah. Okay, so that's all I have. I explained one assignment today, full. If you anyone have any other question, I will try to answer. Otherwise, or if you want me to talk about any specific, what do I say, any specific concept which you're struggling with or something, then in the next class I can talk about it. You can just write suggestion in the chat box if you want. Or else I will go with the normal. Okay.
These were the questions from last assignments, last year's.
Yes, I guess. I think these are from 2022 probably.
Okay.
So, yeah. So, and the thing is, you will get an idea, it will give an idea, the kind of how to solve this question.
This is very helpful for the final exams also.
Yes, yes. I hope.
Yeah.
Hopefully.
Thank you. You're very clear about the concepts. Thank you.
Thank you. Yeah. Sure. So, yeah, see you next week then. You will having other sessions probably from 7 to 9 today, and tomorrow. I guess 3 to
We will be having how many sessions, like interactive sessions? Because this is a 4-week course, no?
Yeah, it's a 4-week course,
Yeah,
basically.
so we'll be having some more sessions next week also.
Yeah, so next week, so 2 more weeks are left for
Okay.
my class. But in total, you will be having 12 classes because I mean my other fellows will also be taking.
Okay. So basically, till the exams, there will be some online sessions which are going to happen.
Excuse me, sorry, what is the question?
No, basically, I think till the exams is sometime in March, no? Till then, there will be, even after the 4 weeks lectures are done, we will have interactive sessions to get our doubts solved.
Okay, could be. We can clarify that.
Thank you.
Yeah, yeah. So I will upload these videos on the YouTube. So at the end, I will I have not uploaded yet. So once I upload it in the YouTube, I will share the link here. Okay, thank you everyone. Have a good day.
Thank you, ma'am.